So in the last 20 minutes before lunch, we're going to take a giant leap into the molecular guts of biological systems. So I'm going to talk about computational modeling and what kind of capabilities we have there. So computational modeling has become an integral part of many research projects. And one of the objectives of the molecular science computing facility at EMSL is to support these activities by providing an integrated computing environment that combines together the advanced high-performance computing resources in the form of a supercomputer, data archive, and also the software development. This really gives you a unique environment to integrate simulation into the experimental activities and to address problems that would be very difficult to do otherwise. So the software that I'll be talking about is NWCam, and it's been around for quite some time. It started, the original development started as part of EMSL construction project in the mid-90s. And been growing steadily since then. We have about 40,000 downloads in the last 18 months. So the key features about NWCam is that it covers a wide range of chemical systems, starting from gas-phase molecules all the way to biological systems. But that offers a lot of advantages in the form of the problems that you can do. NWCam is also known for very high efficiency on high-performance computing platforms. So in this talk, I will talk about how NWCam can be applied to a biological system. So, as I said earlier, we are going to the molecular level description of biological systems. So, we're going down to atoms, including electronic structure. So, the importance of this type of investigations really lies in trying to understand the chemical transformations that drive the biological processes. It's important for de novo protein design, when you try to construct the biomimetic systems, and also in the interpretation of experimental measurements, NMR, etc. With this level of detail, the molecular level of detail comes a price tag. So these type of calculations are usually very computationally expensive. Another aspect is that you tend to generalize the data. So you have to be careful about answering the problem that you want. So when it comes to molecular level descriptions, you really have two methods. One is based on quantum mechanics, where you solve the Schrödinger equation. Another one is a classical molecular mechanics description, basically bolts and springs. Both methods have their pros and cons. Obviously, the main advantage of quantum mechanical methods is that you have explicit description of electron degrees of freedom. But because of that, it's also become computationally expensive for large systems. Molecular mechanics, on the other hand, is very efficient for protein simulations and really the method of choice there, but it cannot describe chemical reaction. This information is simply not there. And also, every time you encounter non-standard cofactors or different amino acid residues, you have to parameterize the force field. So in NWCam, both methods are available, and both can be used for biological systems. But the presence of the two modes of description on the same software package also offers a different approach, where you actually combine the two methods. So if you think about it, do we really want to use quantum mechanics to describe the entire protein complex? Probably not, because the chemical identity of the protein really remains essentially the same during the course of evolution. But there are small regions there, the active sites, where the chemical changes take place. So it's natural to try to restrict the quantum mechanical description to the regions where it's important and use the molecular mechanics for the rest. So this gives rise to a QMM methodology, which you may have heard about. So by using this combined or hybrid description, you gain immediately in efficiency, and you also you also achieve some data reduction that makes it easier to interpret the simulation. So I'm just going to go what were the current capabilities of QMM methodology and WCAM, which include structural characterization, property calculation, study of reaction pathways, and free energy calculation. So what I mean by structural characterization is that you have a region in your protein complex or your biological system, and you want to really get a high precision structural information from it. And the method that we use is a combination of molecular dynamics and optimization. So typical use cases for this type of simulation would be if you have an X-ray data, which is probably not of high quality and you want to improve it. Another application would be if you want to generate a structured protein complex which has a different cofactor or residue, and that's frequently the case for enzymes because active enzymes are active, so you cannot catch them. So you do some various modifications to freeze them in various states. You can also generate structures of various intermediate or product state complexes where you force the system to break certain problems. So and again, these structures will not be available experimentally, so you gain some new information. And here's the method that we use. Really, the thing you have to understand about this is that by using this methodology, we reduce the time to solution using conventional protocol from several weeks to a few hours. And that's a big deal for us. So you can take a protein structure and you can refine it in a matter of hours. So I'll give you some examples of this type of applications. So first one is the CMP-dependent protein kinase. So the original X-ray structure 1-ATP contained a peptide inhibitor complex. So we wanted to see what kind of changes would be, what is the reactant state of this protein with the active substrate complex. So we mutated the alanine residue, which was present in the inhibitor complex, to serine, and subjected it to the activation process. And the outcome was the structure. So you can really learn about this. Don't have time to go in details, but one important feature you have to notice is this contact between the aspartate 166 and the serine substrate. You can see there's a nice hydrogen bond here, which will be important later on. As I said, you can also generate various product state complexes of your protein structure. Here, we take again the CMP-dependent protein kinase and looked at the product state. And the way we obtained this product state, we forcefully broke the bonds, the PO bond in the ATP molecule, and we transferred the proton from serine substrate to the D166. So this turned out to be a stable complex. It is a product state complex. And again, there's a wealth of information here. One important observation is this. The magnesium ion, which is coordinating this bridging oxygen on what used to be an ATP. So that's an important thing. There's also other things here. There's one more example related to ribonuclease A. So here the starting gas came from the RNase A by the vanadate complex. So you had a vanadate instead of phosphorus. And so what we did, we replaced the vanadate by phosphorus and we optimized the structure. As you can see, the backbone really didn't change much. So the green is the computed one, and the blue is the X-ray. But we have some significant changes in the active site. So the original X-ray structure here is exhibited by lines. In particular, if you look at this lysine residue, it really shows a different conformation. And that's again important for the reaction process that I'll show you later. In addition to structural characterization, you can also perform a variety of property calculation on protein structures. This could be NMR, EPR, UV, Raman. We can investigate both static and dynamical structures. And by comparing the computed signals with the ones with experimentally measured, you can get some information, or the opportunity to get the information about the local environment of the protein structures. So you're really extending the scope of your experimental measurements. So this is an example of the NMR calculation that we did for zinc metal protein group redox. So as I said, we can perform simulations or the property calculation not on the static structures but on a dynamically evolved one. So this is an example calculation of the strand of DNA, and we looked at the UV signal of excited states. So we looked at the first two excited states, and the important thing here was that during the evolution, these two states never crossed each other, so they remain very well separated. And this has an implication for the photostability of the DNA, meaning that when the DNA gets excited, the first state can decay radiationally into this sort of harmless structure, while the second can lead to all sorts of nasty things. Another thing one can do is calculate redox properties. Here's an example calculation of the cytochrome and the sugar bacteria. So it's got 4 iron cube centers, and we can calculate the redox potential for all 4 of them. And that has an implication for electron transfer processes. You can also analyze hydrogen bond interactions in the active site. The way to do it is you look at the OH or NH bond stretch frequency and see how much it's shifted compared to the vacuum. So the more it's shifted, the stronger your hydrogen bond interactions. That also gives you an opportunity to study what kind of networks do you have in the active sites. So in addition to studying reactant state and product state complexes, we can also look at the reaction pathways. So basically, this allows us to map the reaction pathway between the 2 stable points, the reactant and the product, and gives you an opportunity also to get the transition state. So, here's an example of the reaction pathway calculation for the CNP-dependent protein kinase. And so this is the structure of the transition state, and the important thing to notice here is that the substrate proton still remains in place, even though the PO3 group has moved quite a ways. It's sitting right in the middle. So this was an important thing because prior to that it was known whether the substrate proton is a late or early event in the reaction process. So the product state is proton transfers to the this catalytic ASP. So here is the transition state structure that we obtained during this calculation. So as I mentioned, the substrate protons to it. Another important thing that we found out in the course of this investigation is the role of this secondary magnesium ion. What happens in the reaction process is that this magnesium ion plays an important role in coordinating the development of this negative charge on the AGP as the PO bond is breaking. So that's an important thing. This magnesium ion is known to be very important in the catalytic activity of this enzyme. Here's another example of calculation. So this goes back to the RNase enzyme. So what was interesting in this calculation is that originally it was presumed that the histidine 12 would be the species that donate the proton during the hydrolysis. But it turned out that actually it's a lysine, which was very unusual. And once you have a reaction profile, you want to know what are the energies associated during the reaction. So it's actually a very difficult problem to do quantum mechanically because you need to do a lot of sampling to get the free energy differences. So we use, the algorithm we use is based on the thermodynamical cycles where we use an effective classical representation, and we go around using thermodynamical cycles. Basically, we shift the statistical sampling from higher level of theory to lower level of theory, and then correct it. So for the protein kinase reaction that I mentioned earlier, here we have the resulting reaction profile, and we can see that the activation free barrier is around 50 kcal per mole. The experimental and measured one is about 13. So there is some predictive quality here which can help you to study the catalytic activity of the enzymes. And for RNase A, we get this following profile. So again, here's the stable intermediate complex, and the reactant, and the product state, and also the transition state. So I'm going very quickly, but basically, I guess what I'm trying to tell you is that there is a lot of power in quantum mechanical simulations nowadays that can really help you to answer some of the questions that might be interesting in biological simulations. So let me end up with some perspectives, the stuff that we do with NWChem. So we pursue new methods for multi-scale, multi-physics simulations that will help us to study bigger, larger systems and get the answers that we want. We also want to really driving towards the user-friendly experience, because these calculations are complicated. Doing complicated things and calculation setup could be somewhat formidable for a non-expert user. But when you do these things over and over again, you know there's a certain pattern, certain templates one can use. So what we're trying to is the sort of remove this mundane task of constructing input files and then jobs on the supercomputer and sort of hiding from the user in some way. So this allows you to sort of concentrate on the science rather than these things that are not really important to science. And finally, this also provides an interesting way of integrating the simulations with the experiment. Where, for example, you take the NMR measurement and you hook up directly to, say, simulations that can start interrogating various candidate structures, see if you can match the NMR signal, and it gives you some new interesting information. With that, I would like to finish. Thank you for your time.
Other questions from Marat? So have people started to use some of these predictions then and gone back and put those mutations back into proteins and close that loop between what you predict and what actually happens? Or, I mean, is there a possibility of mutation?
Oh, yeah, absolutely. The thing is that what I find with, there's a certain barrier, you know, everybody talks about genomic cell signaling, and then you come in with molecular level description, and there is a certain barrier people sometimes do not actually believe you can do that. And yes, actually you can. So, for example, in the case of a kinase, you can start mutating different residues and see what kind of effects you get. And it's a fascinating area. The one that hasn't really taken off yet because of demands on the computational time and also some know-how on how to do these kinds of simulations. But my feeling that it's going to become more and more and more important. And I'm particularly excited about the coupling between, say, NMR measurements and theory, because really there you really have the opportunity to gain sort of much more from a single thing just combined.
Okay. Oh, another question. Does the software, specifically the user interface, work better on any platform, or does it work equally well?
It works equally well on anything starting from the MacBook to a supercomputer. All the major supercomputing centers have Untitled and actively support. Obviously, the big calculations you do not want to run, on the personal machine. And obviously, that we can run very well on Chinook. The nice thing about Chinook is also you get really experts